Hello, I'm Lynn Morris.
And I'm Penny Moore.
And we are research scientists based in Johannesburg at the National Institute for Communicable Diseases. South Africa is the epicenter of the global HIV epidemic, and a vaccine is an urgent priority. This is especially true here, where in some areas up to half of young women are HIV infected. A vaccine will likely require the elicitation of what are called broadly neutralizing antibodies. These types of antibodies are able to block infection by diverse viral strains from across the world. This is a particular challenge for HIV, which is a highly variable virus. Animal studies have shown that if broadly cross-neutralizing antibodies are present before the animals are exposed to the virus, they are protected. However, despite years of research, we have been unable to elicit broadly neutralizing antibodies by vaccination. Encouraging studies in infected humans have shown that a minority develop broadly neutralizing antibodies that we hope one day to elicit with a vaccine.
Understanding the pathway to neutralization breadth in these HIV-infected people has provided useful insights into exactly how broadly neutralizing antibody lineages emerge, likely a combination of both viral and host factors. We have known for many years that high viral loads contribute to neutralization breadth, but what we're now understanding is that how the virus evolves is just as important. In this review, we focus on the role of viral characteristics and evolution in shaping broadly neutralizing antibodies during HIV infection, and describe how these findings can be translated into novel HIV vaccine strategies. This has really only become clear recently with longitudinal studies that trace the emergence of these antibodies, shown here in the red wave, from their strain-specific precursors in orange. New technologies have improved the isolation of monoclonal antibodies, and we can use deep sequencing approaches to trace back in time to look at the unmutated common ancestor. As shown in the bottom panel, parallel virological studies have allowed the field to define the viruses that initiated the broadly neutralizing antibody lineage, and the subsequent viruses that drove the antibodies towards breadth. This is a cat-and-mouse game where the virus changes, and the antibodies then change to keep up with the new viral variants. Work from several research groups over the past 5 years has highlighted at least 3 mechanisms where viral evolution contributes to the development of neutralization breadth. These are described in detail in the review and summarized in this figure. Viral escape can result in the creation of epitopes that become the target of later broadly neutralizing antibodies. There are also examples where viral escape from an early antibody forces the virus to expose an otherwise hidden epitope. And finally, there is a lot of exciting data coming out to suggest that as the virus tries to evade early antibodies, it attempts several escape routes, creating multiple epitope variants. This diversification results in early antibodies learning to tolerate variation within their epitopes. In a sense, the antibody swarm becomes more promiscuous and is able to recognize many more viral variants, and that's what allows these antibodies to become broadly neutralizing.
Of course, understanding this process has very practical implications for HIV vaccine design. It suggests that vaccine strategies may need to replicate viral evolutionary pathways. Such a sequential immunization approach would coax the immune system towards breadth. Overall, understanding viral drivers of breadth described in this review has provided many useful insights that could be harnessed to develop an effective HIV vaccine.